Okay. Welcome to Get Selly with It. During this webinar, all participants will have their mic and videos muted. There will be a question and answer session at the end where Dr. Clay will use that time for questions submitted through the chat feature. We will allow participants to come off mute and ask their question if specified within the question that's what you'd like to do. My name is Sydney Stern. I'm the Director of TGCT Programs at TGCT Support. I will be your moderator for today. We would like to thank our sponsors, EMD Serono, Decipher Pharmaceuticals, and Daiichi Sankyo, and thank you all for spending your evening with us. So now I would like to introduce the very impressive speaker today, Dr. Michael Clay. Dr. Michael Clay is a dual board certified anatomic and molecular pathologist with service responsibilities in adult and children's hospitals at the University of Colorado and Schwitz Medical Campus. And Dr. Clay directs the bone and soft tissue services at this hospital. He also has service responsibilities as a molecular pathology group at Children's and many other accolades. And so we're very grateful that you'd choose to spend your time here. I think many of us have questions, and I think I sent some of them. So we'll just get started, and I'll let you go ahead. And at the end, we will take questions. So please feel free to submit them in the chat feature below during the talk.
Great. Thank you. Hopefully everybody can hear me okay, and hopefully I'm sharing the correct screen. Just let me know if something goes amiss as we go forward. Um, as the kind introduction mentioned, I'm a director of bone and soft tissue path here at the University of Colorado. And I'm also a molecular and genetic pathologist. So I have sort of unique skill sets that sort of overlap most of the areas that matter in diagnosing tenosynovial giant cell tumors, which is what we're going to talk about. All right, so I'm going to have a brief overview. I'm going to talk a little bit about some definitions. As many of you know, this is a complicated disease type. It's very confusing in the literature. There's a lot of changes that have occurred recently. I'm going to talk about the molecular findings because those are all really important and definitely a hot topic for patients undergoing sort of targeted therapies. Talk about histology, and that's what I see in the microscope when I look at these tumors, and then talk a bit about the natural process of this disease. Fortunately, I was given a small list of patient questions ahead of time. And so as I go through my talk, I'm going to highlight some of these questions and answers when I come across them. So hopefully, if you've submitted a question, I'll be able to answer that for you. And if not, write it down and I'll definitely cover the things when we're done. I thought I'd start first by saying who I am. What do I do? How did I get where I am? Just to give you sort of a context for the things I'm going to be saying and why maybe you should listen to me if you've got a question. So I'm a pathologist, and that's a physician who studies diseases and their causes. Primarily, where patients are concerned, I make diagnoses. So I thought I'd show you my journey just to sort of give you context for how I got where I am. A lot of people don't really understand sort of medical school and how all of that works, and I thought I'd give a quick rundown of the different things. So I did undergrad. I'm from Maine. And so I did my undergraduate training in Maine for 4 years. Then I went to med school, went down to Vermont. Usually med school is 4 years. I did 5 years of med school, which included a special year of pathology. It was sort of a way to get more experience with path and help me launch into a better program for residency. So I did that. I went off to Stanford, California for 4 years of residency. Then I did specialized training just in bone and soft tissue tumors. I did a fellowship in that in Georgia for a year. Stayed in Georgia, did another year of molecular and genetic pathology fellowships. And then after that, I took my first job. So I worked at St. Jude Children's Research Hospital for a number of years. A lot of kids get sarcomas, and that's my expertise. And so that was a good first job. And then I went to the University of Colorado, which is where I am now. As far as you're concerned, sort of practically, a surgeon will be the person that takes a tissue off of a patient and they send it to somebody like me. So it goes from the surgeon to a pathologist, and a pathologist tells them what it is. And then we have sort of advanced discussions. All right. So a little bit about how it works, just because a lot of people haven't seen slides like this before. So let's pretend you go to the operating room and they have a little bump. You get a little nodule taken off. It looks like this. So that gets put in a jar and it comes to pathology. And so what happens then? Well, it gets put in very special fixatives that sort of cross-link the DNA and the proteins and put the tissue sort of in stasis. That tissue is then cut in a certain way that we can reorient. It's put in these little plastic cassettes. Those get processed, and then they get flooded with essentially what's like candle wax. And so imagine your little tissue gets a section cut out of it, and now it's embedded in candle wax. And why do we do that? We do that because it lets us cut it super duper skinny into these very thin ribbons, which are then floated on water because it sort of makes things easy. And then you use a glass slide and you pick up one little section that has been floated, goes through our complicated staining system, and then I can look at it under the microscope. So that's sort of generally how processing in histology works. So all of your tissues will have started like this, gone through this same process, and you have slides from your tissue stored somewhere, and in many places stored forever. Okay, so definitions. Tenosynovial giant cell tumor, which you might see referred to as TCGT or TGCT or TSGCT. These are a group of lesions that most often arise from the synovium, usually in joints, but also can happen in bursa and tendon sheaths, and they show synovial differentiation. TCGT can be classified into intra-articular and extra-articular subtypes clinically. So, like in the clinic when you're talking to your doctor, they can be classified histologically, and that's with a microscope, into diffuse and localized subtypes. And although diffuse and localized have the same molecular signature, we consider them the same type of disease process. The clinical context that they present and the sort of behavior in a patient is very, very different. And so it's very important for us to be able to distinguish between these two things. Some of the related terms you might have heard, so pigmented villonodular synovitis or PVNS, that's what these were called maybe 10 years ago. That terminology is no longer appropriate, no longer used. Or giant cell tumor of tendon sheath. These are sort of the old terms for localized and diffuse types, respectively. We don't use these anymore. Okay, so let's talk a little bit about these two different subtypes, just sort of generalized basics. So the localized subtype is lobulated. They're usually very well circumscribed. They're relatively tiny, 2 to 3 centimeters. We're going to go over cell types, but they have sort of a lot of cell types in them. And they're usually cured with simple excision. The diffuse type is very infiltrative, meaning it grows into other things. They often grow as diffuse sheets under the microscope. They can be quite large. 3 to 13 centimeters is the average. They're more monotonous, meaning there's less of this diverse cell type population. And these will often come back or what we call recur. So first question I got from one of the patients is why do pathology reports say diffuse type, TCGT or TGCT, when the MRI says localized? That's a super good question. And it comes down to the criteria used. And so diffuse type is a histologic diagnosis and a gross tissue diagnosis. You have to be able to see the tissue growing into other things. You have to be able to look at the edges of it. You'll see that the small ones are 2 to 3 centimeters and the larger ones are 3 to 13. So if you imagine if you get one of these tumors that's exactly 3 centimeters, say, an MRI might look at it and they might just call it localized because it's tiny. But if I get it and I see it's growing into a tendon or something, it's definitely diffuse subtype. And so MRIs ought not to say that something is a localized because the criteria are histologic and gross, and you can't do that with an MRI. You can guess. I mean, there's correlations, right? The small ones are usually localized, but you really need a microscope, which is why sometimes the radiology and the final diagnosis might change or vary. All right, so I'm going to show you some photos of these things because I think it's really helpful to understand the context. So there are some, surgical pictures coming, some tissues. So just be aware, look away if you need to. So here's what a localized one looks like. Like I said, lobulated, well circumscribed, and tiny. You can see this bump in this patient's finger. When the skin is opened, it's a very well-defined, lobulated, and marginated sample. Right? You can just take your finger or whatever, and you can sort of draw along the edges of this thing. And when it comes out, you can see it's got a very sort of clean edge with the surrounding fat. This is the tumor here. And this sort of clear stuff you can almost not see. That's what fat looks like under a microscope. And so this is all fat, and this is the hard bump, which you see here. It's obviously very circumscribed, meaning it has a firm edge at one side. And why does that matter? That's because a surgeon can follow that, right? This fat stuff cuts just like, when you're, cleaning up a steak or something, right? The fat is very soft and easily separated from the meat. In this case, the fat is very easily separated from the tumor, and so it's easier to cut around and it comes out entirely most of the time. What about the diffuse ones? So these are actual photos of diffuse type tenosynovial giant cell tumors, and you'll see that they're very large and they're sort of irregular and sort of shaggy. And so like this one has a very sort of ragged edge um, at the side. And the question would be, where does this tumor stop and where does your healthy tissue begin? I can't tell by looking at this thing. It's super complicated. It's all giant, it's frondy, there's a bunch of fibrosis. I'm sure there's some normal healthy tissue in here somewhere, but I can't tell. And neither can a surgeon when they're trying to remove one of these things. And so you often get the tumors cut, and thus they recur. It's important to note how large this particular one is. That last little tumor I showed you was in the finger. This is what it looks like to scale with this other one. So this is a giant tumor from somebody's knee. This was that little one from somebody's finger. And so just really large, irregular, hard to sort of get around. So histology, that's what it's called when we look through a microscope. We look at the histologic features. So specialized pathologists make these diagnoses pretty regularly based on just slide review. Um, we don't routinely do molecular testing, at least in my group. And it's a very rare situation that ancillary testing may be used, but there certainly are exceptions. I'm going to run you through what they look like just because they're very interesting looking tumors, and it gets to some of the maybe more pertinent questions about your molecular testing. And so this is a picture. I took this picture through my microscope here, which you can't really see, but this is a picture of a tenosynovial giant cell tumor. And there's a lot of stuff going on here. I'm sure to many of you, it looks like dots and squiggles and colors. So I'm going to highlight just some things. There's at least 6 different cell types in this picture. Most tumors are pretty pure. So if I took a section of like a squamous carcinoma, like maybe off somebody's neck or something, it's almost all tumor. And so these lesions are very unique and they're super mixed up. And so these are foamy macrophages, these big bubbly looking cells. Those are reactive. We've got these giant cells, and that just means that the cell has many, many, many nuclei instead of these little tiny ones. Um, and they're sort of haphazardly scattered about. Those are also reactive. We've got fibroblasts, these little skinny ones here. If you think about like a scar, if you get like a scar on your arm or something and it sort of gets all hard and weird, these are the cells that make scar tissue. So they're in here sort of making things scar up. We've got lymphocytes all over the place in here. Lymphocytes are part of your immune system. So if you get like a virus, your like lymphocytes will be fighting those things, but there's lymphocytes scattered in this. Those are also reactive. I found one little red blood cell at the bottom, this little tiny red thing. And so that would normally carry oxygen through your body. I'm sure it's carrying oxygen through this tumor, but there's definitely red cells also floating in and out of here. The tumor, just like your body, needs oxygen to survive. And so it has blood vessels and all the things that your leg would have, for example. And the tumor cells are hard to identify. I would say these larger single mononuclear cells are good for tumor cells. Sometimes you get sheets of these things. There's just like tons of them back to back. And sometimes it's more like this, which is just a hodgepodge of things that are thrown together. I thought it'd be fun to show you a real example. So I'm going to click this link, see what happens. And it has pulled up a slide. I'm going to slide it over. Please shout out somebody if you're not seeing a whole slide image right now. So this is a scanned section of a tenosynovial giant cell tumor. And so my job would be to look at tissue like this all day long and say what it is. And so I wanted to show this just because those high power photos I was just showing you, I think are a bit misleading as far as, the scope of what we're doing. And so I can zoom in on this. This is a digitally scanned slide, and you could see there's a bunch of these little mononuclear cells. This one is much more monotonous than that last one, but this is sort of what that high power field looked like, something like this. But when we look at these tissues, we're looking at a ton of tissue. This is one tissue block. So that little thing embedded in the wax that I mentioned, this is just one of those cut. And it has all of this tissue in it. And so someone like me is very specialized at sort of hunting around and looking for important features in here. And here's some of those bubbly foamy cells like we saw in the other slide I just showed you. And so you can see a little bit of similarity between that case and this case, which are from different patients. Um, Way over the side here is a little bit of, it looks like tendon tissue. And so there's just a lot going on. I thought it was just sort of fun so you could see, this is, I might look at 20 slides like this if you had a large tumor excised. Um, you can imagine how much time and sort of effort that would take to do well, and et cetera, et cetera. All right. Hopefully you're back to the slide presentation. Just stop me if you're not. So there are some stains we can do that'll highlight the tumor cells. Like I said, it's sort of hard because most of the stuff in there is reactive. This is a good one. This is a clusterin stain. And so unlike that last slide I showed you, which was like pink and purple and whatever, we can also stain it just this really pale blue color. And then we have special markers that'll pop brown if they stick to it. And so in this case, this is a clusterin stain. Everything in the body would stain to sort of bluish color, but only the tumor cells would highlight with this brown stuff. And so you can see, if you look at sort of count the dots, there's a lot more of these sort of blue things that didn't pick up any of the stain versus these few larger brown ones. And so this is just sort of a nice visualization of the brown ones are the bad ones doing the thing, and everything else is sort of reactive. So going to the molecular a little bit. Um, Tenosynovial giant cell tumors have a pathogenic gene fusion between two different genes in your body. In this case, the CSF1 gene and one of the collagen genes, collagen 6A3. So one of the patient questions was, what causes TCGT? From a more sort of holistic standard, nobody really knows, right? I mean, you might get unlucky in your life and get a number of different tumor types. Sometimes we can correlate those with certain exposures. So you think like asbestos gave everybody mesothelioma sort of back in the 70s, and a lot of people in the military get exposed to really horrible things, and they can get certain tumor types and leukemias and things. We don't know the thing that kicks off this tumor. It might be bad luck. It might be something we don't know. Um, we don't know what causes this gene fusion, but we know that this gene fusion is the thing that then causes the tumor. Um, and so it's a rearrangement between two different chromosomes. These two genes are on different chromosomes in your body, and they come together to make this new product that makes the tumor. And so traditionally, again, I'm a molecular pathologist, so I think this stuff is important to understand a little bit. Traditionally, if you think about like a gene fusion, there's a lot of tumors driven by gene fusions. Um, Normally, that would be one gene in your body comes together with another gene in your body, and you get some sort of like Superman gene that's a mixture of the two things and does something bad or something hyperactive or something. That's actually not what is going on here. We still have a rearrangement between the two genes, but it's a little different, and I'm going to show you how. This is just showing you sort of how we detect these things in a molecular lab. And so we sequence the DNA of your tumor. And these are nucleotides in the tumor, A, G, G, C, C, whatever. Um, we map that, and this says, okay, this bit of DNA came from this part of the gene. And we're going along, and suddenly it doesn't match anymore. We're like, okay, well, it doesn't match. Maybe something went wrong. Like, oh, okay, no, definitely it doesn't match. This part now maps to this other gene. And the system will spit out an output that says, hey, we have a gene fusion between these two genes. The DNA is comprised of both parts of the genome. The the fusion event that happens in tenosynovial giant cell tumor is different. Um, I'm not going to go into super detail, but it's important to understand that it's different. And it explains why sometimes if your tumor gets sent out for this test, it will not come back positive, even if this thing is present. And it's because of the odd nature of this type of fusion. So here's the two genes that we're talking about, right? CSF1 and this collagen gene. Um, all genes have promoters, and then they have the part that codes, the thing that does the thing. And so when this gene is doing its thing in your body, special stuff comes down and it lands in this promoter region, and then it makes the gene product. And that's how it works. And all the promoters in the different genes are a little bit different, right? Like growing hair happens all the time, whereas maybe growing your fingernails happens slower. So one has a really strong promoter and it has a lot of stuff going on, and one doesn't. So it's just important to know that different genes have different things that drive them at different rates. So in this case, the CSF1 gene is relatively slowly promoted in normal situations. The CSF1 gene does have a lot of normal functions. We're going to go into that a little bit later, but it's not an overly expressed gene. So the analogy would be this like little clunky train. It has its thing, it goes together. It's just sort of slow in the product that it makes. The collagen genes are very highly promoted. Collagen is like in your skin and it's sort of These are a lot of maintenance genes about like healthy sort of turnover of your tissues. So I would liken this to sort of, this monorail train, much more fast. It's much more fastly going. So the odd thing that happens in this fusion is we don't take one gene and stick it on the other one. That's actually not what's happening here. What's happening is we're taking this promoter, this part that's outside the coding region, where all the fun machinery comes down. We're taking that part off of this collagen gene that's super active, and we're sticking it right on the front of the CSF1 gene. So what do you get? You get this new engine blasting a super high level of CSF1 that normally wouldn't be done. Hopefully that makes sense. Um, so unlike a normal gene fusion where you get like one gene stuck to another gene, sort of like Spider-Man hybrid of you create a new product. In this case, all of the normal CSF1 is there. We just changed the engine in front of it essentially. Um, why does that matter? It's interesting, but when we sequence DNA or RNA for detection of these things, usually we just care about this part. We care about the coding part because 99% of fusions are this, coding part becomes sort of a hybrid with another one. And so these promoter regions are often not sequenced. And so unless your lab is specifically looking for it, you order this test, you can sequence CSF1 all day long, but if they don't include this spot, you'll never see that it got cut off and this other thing got stuck on because the sequencing would end right here at the beginning of the gene. Hopefully that makes sense. So like I said, it's a hard to test for, because it's a what's called a promoter swap. If you wanted to Google that later, that's what this thing is called. And we often get false negative results when we send for it. And it's not just because of that weird fusion. It's also because of all that background stuff. So remember, the tumor cells are a minority. So I'm going to liken it to this little diagram I made. So this little green thing is your tumor cell, those things I pointed to with the arrow, and these blue ones are just all the other ones in the background. So how does a molecular test work? Well, we take the DNA out of all these cells and we look at them, but it's all at once. And so you might think we take this, we blend it up, and we have some of your normal DNA and then this little bit of tumor DNA in the bottom. Um, Conceptually, that's true, but that's actually not what the machines do. And so instead, you really get a blender. And so all of this stuff gets mixed up together, and the molecular machinery, the testing that we do has to say, hey, do you think this is blue or do you think this is green? It's really all these things often come down to sort of signals. Is the signal blue or is the signal green? And so in this case, we can all look at this image and I imagine we would all say it's blue. That's what the machinery is doing too, and it's going to miss the fact that this green thing is there. One fun thing I did is I actually took this blue and I overlaid it on this green color. So in this image, this is 100% of a blue color, and this is 5% of this green color coming through the background. And so this, you're looking at this color right here. This color is actually 5% green and 95% blue. I actually sort of did that with the software. So these two colors look pretty much the same to me, even though I know for sure this is actually 5% of that green color. And that's what the machinery is doing. It's trying to guess, and it gets it wrong. There are ways we can sort of get around that, but that's one of the reasons why your testing might come back negative. Hopefully that makes at least a little bit of sense. So there's some other tests you can do. Again, these often are not needed in most cases. So just in case you haven't heard the terminology, we can do IHC staining. Again, I showed you that stain earlier. Again, there's one for CSF1 where the tumor cells that are making that turn brown. You can see that here. And you can also do this fancy hybridization study where we put like fluorescent probes on top of a gene, and then if that gets broken up, we see the dots get sort of separated. And so normally, and imagine we're going to put one red probe right here, one green probe right there. If I look at it under the microscope, they're going to be touching because that gene is like all wrapped up. But if this break happens, whatever part of CSF1 that was here gets spread off somewhere other else in the genome. And so they get separated in space. And so you can see that with these probes. That's what's happening right here. This is normal. Red is right on top of green. And in this case, there's at least one copy of this gene in here that got broken half. That would let you know something happened to the CSF1 gene. It got split. So I'm going to go a little bit into sort of what happens then. I'm not a molecular biologist. I'm a molecular pathologist. So This isn't really my realm, but I thought I would show you just a little bit about, okay, so now we have this fusion, then what happens? So very basically, here's a tumor cell. So the tumor cell makes too much CSF1, right? Remember, it's got that new rocket engine on front of it, so we're just pumping out a bunch of it. So step one is the tumor cell's making too much of that stuff. Some of the cells in your body normally have receptors to CSF1. It's a real protein. It does real important things. Some of your cells have that receptor, and they get recruited to this location because of all the signals getting put out. What's the result? Well, you get a recruited landscape of mostly reactive cells and then relatively few tumor cells. It's obviously a lot more complicated than that. Some of the recruited tumor cells also secrete rank ligand. That's just sort of another chemical pathway in your body. And that has much of the same factor. It recruits other cells, it gets sort of a feedback loop, and it activates a lot of different pathways in your body. Um, that's why there's a lot of symptoms that go along with having these tumors. It's not just I have a big bump, but you get joint swelling, you get bone and cartilage destruction and remodeling and stuff from all these pathways getting activated. A lot of these things can be targeted with drugs, and so I'm going to talk about that a little bit. Another patient question. Is there anything else abnormal with TCGT besides the CSF1? Um, that's a hard question. I would say the CSF1 fusion is the thing that makes this a tumor. It's the thing that drives the whole process. So that's what causes the tumor. Sometimes there's other things you can find. And so sometimes there's what we call copy number changes. And so sometimes you have gains of certain chromosomes. So you can have a gain of chromosome 5 or a gain of chromosome 7 in the tumor. Those are secondary to this CSF1 event. Um, some tumors also get secondary mutations. And so CSF1 starts, that drives the tumor. But like every cell in your body, it's allowed to sort of have other things happen to it. And sometimes they pick up mutations. And so P53 is a tumor gene, and sometimes that gets mutated in these tumors. When that happens, we can detect it with sequencing, but it means other things. Like those that often correlates with tumors that are more aggressive or might be displaying sort of progression to a more sort of aggressive phenotype. But you know, this CSF1 is what causes these things, period. And then sometimes there's other things that can be identified. So targets. I got this slide from Dr. Wilkie, who's one of our oncologists that treats people with TCGT. And this is a really cool scenario. We have a lot of tumors. I'm a sarcoma person. I diagnose soft tissue tumors. Many of our tumors that I diagnose are horrible. They're based on gene fusions that are not targetable, and people get really toxic therapies. There's not like a really sexy pathway that has all this cool stuff that's been studied by scientists that can be, have drugs designed toward it. So this tenosynovial giant cell tumor having this very well-known pathway and having drugs that are characteristically like designed specifically to target those different parts is really unique in medicine. There's a couple other examples, but this is really sort of amazing and very unique. And so you can target this whole process in 1,000 different locations. And so there's this drug here that actually is an antibody that binds the CSF1 itself. So your cells make a bunch of it, and the stuff just sort of gobbles it up. Um, you can say, well, okay, fine. I'm going to gobble up the receptor instead. Um, so this is sort of a weird analogy. If you think about like throwing a baseball and catching it, you could grab that baseball out of the air and then it can't be caught. Or you can block the glove and it can't be caught. And so there are drugs that block the glove. Some of the drugs attack the ability of that receptor, or in this case, glove, to exist at all. And so in this drug would block it so the glove can't open, right? It can't do the thing it's supposed to do. So you can really block this process in a lot of different locations using these really clever design drugs. Um, these pathways, these drugs that target these certain pathways have a lot of overlap with other pathways in your body, which is why you get some weird symptoms and why some drugs designed for other tumor types might also work in this tumor type because there's a lot of crosstalk. It's just sort of above my pay grade. But you know that in medicine, this is a very unique biological process, and it's sort of fascinating that the drugs can exist. It's really not like that in most tumor types. All right, is CSF1 involved in any healthy process or other diseases? That was a good question. Um, I took this directly from the internet. So I went to this website, genecards.org. I go to that website a lot as a molecular pathologist because it tells me about genes. if I'm looking at a mutation in some patients, and I'm like, well, what does that gene even do? I like to go to this website. It gives you a lot of detail, but if you just look up CSF1 gene, it says it's involved in the differentiation of those macrophages, which are some of the cells like the foamy macrophages I showed you. Um, it is involved in inflammatory chemokines, sort of an inflammatory reactive process in your body. Um, it's an important role in osteoclastic proliferation. Osteoclasts are the cells in your body that make bone, and that's probably why when you mess around with CSF1, you end up with a lot of changes in the bone because normally it's involved in that process biologically. It's also required for normal male and female fertility. I don't know if this disease is associated with any abnormalities of fertility. Um, I learned this bit when I, learned this. But it does a lot. It does a lot of things, which is why when you mess with it with a tumor like this, you get a lot of weird symptoms, and a lot of the symptoms are not the same in every patient, right? We all have sort of different genetics, and our body's a little bit different, right? Like I have hay fever, you don't have hay fever, my body reacts a little differently. And so everyone's CSF1 is a little bit different, and that sort of explains why some people get really bad joint disease and some people don't have any joint disease, and sort of everyone's a little different. And so that's why your symptoms might be different, but also your reaction to the drugs might be different because everyone's system here is a little bit different. But answer in general, yes, yeah, the CSF1 does a lot of stuff, um, and it's sort of all over the place in these pathways. So clinical implications. I thought I would mention this just as an aside because I think it matters. So what happens when a doctor biopsies your tumor, your tenosynovial giant cell tumor? What does that mean for me diagnosing it? It's important to know what that means. So like if we look at this original case, I showed you this little thing from the finger. This is that tissue. So what is a core biopsy? Well, core biopsy, we have like these needle guns and they line it up and they fire it and it shoots the needle down through the tumor, retracts, and I get this skinny little strip of tumor that we then put in the wax and I look at it under the microscope. I told you up above that I need to see the edges and what it's doing and how big it is if I'm going to diagnose it correctly into these different subtypes. We obviously can't do that when you get a core biopsy. You've missed all the things. Like what if over here on the side, it's invading your bone? I can't see that. I just see what I see here. And so when you get core biopsies, if your pathologist is good, you'll get a diagnosis sort of like this. Hey, this is a tenosynovial giant cell tumor. I can't tell if it's localized or diffuse type because I can't see the whole thing. That's basically the summary of what that means. If I do get the whole thing out and it's localized, those are typically curative. They often do not come back. The surgeon gets all the way around it and the patients are cured. If it's a diffuse type, means the margins are positive often. They often come back. And even when the margins are negative, they often come back. So two more good questions. Um, what makes tenosynovial giant cell tumor different from a sarcoma or rheumatoid arthritis? And why was TCGT previously called PVNS or pigmented villonodular synovitis? And why was that changed? Both super good questions. So to understand these answers, you have to understand sort of how we classify disease processes in your body. And so I would say that there's three classifications that matter here. Reactive process. So imagine you get cut and you have a scar. That scar forming is a reactive process. It might be hard, it might be a big bump. It's not a tumor, it's a reactive process to some sort of assault, in this case, a cut. Um, Rheumatoid arthritis is a reactive process. It is caused by autoimmune sort of problems in your body that are sort of beyond my understanding, at least for this talk. But your body does something and it kicks off this very aggressive inflammatory reactive process. There's no genetics in the tissue specifically that cause it. There are some things that might predispose you to rheumatoid arthritis in general, but all the stuff that's happening, all the cells are reactive in nature. Then we have neoplasms, true tumors. Tumors are usually driven by some sort of genetic underpinning. In this case, the CSF1 abnormality. You can have tumors that are sort of benign. You can have tumors that are super aggressive. Um, how a tumor behaves is sort of where we fall on this spectrum. And so, what makes TCGT different from a sarcoma? Well, sarcomas are fully malignant, meaning they can metastasize, they can travel to other parts of your body, It can go from your foot to your brain, or your heart or your lungs. TCGT doesn't have the ability to metastasize. It's a very local, aggressive, horrible process, but it stays where it is. It might travel locally, it might extend in different directions, but it's not going to pop off, float to your lungs. So that's the difference between a sort of benign neoplasm and a true sarcoma. They both these categories might have genetic abnormalities versus RA, that is a reactive process. So we changed the name from PVNS because pigmented villonodular synovitis. Synovitis is a term meaning a reactive synovial process. And so now that we know the molecular causes this, we know it's not a synovitis, it's a tumor. And so the name was updated. All right. A little bit about specialized central review. There's different types of medical centers, and they provide different levels of care. And so imagine, you go to your primary care doctor because you have a headache, right? I mean, primary care doctors are designed to sort of screen you for everything and then escalate to some other person if they need to, right? You might go to an allergist, which might be a secondary care level. If things get really complicated and you need really specialized help, you might go to a tertiary care center for something like an open heart surgery or very specialized care. There's different levels of pathologists, just like there's different levels of sort of hospital system. And so we have generalized pathologists that might work in a community practice or a private practice group. These people are much more practiced in seeing very common things. So if you go to your primary care doctor and you're like, oh, I have a bump on my arm, you might go to a secondary care doctor or dermatologist. They might cut it off, and it might go to a private practice doctor. If it comes back as a very rare tumor or something very complicated, it'll then get punched up to somebody maybe in a cancer center or somebody at an academic hospital like where I function in sort of this environment for more specialized care. For you guys, primary practice people might make a diagnosis of tenosynovial giant cell tumor, but in the end, it's up to somebody like me to confirm that diagnosis. And so it's always a good idea when you have rare things to get some level of advanced academic review. I would say anecdotally, we overturn a lot of diagnoses from this private practice setting, right? It's not, the private practice person's job to be able to diagnose all the things I can, right? I don't diagnose everything. They have to be good at a small subset of everything. I'm exceptionally good at a very small subset of one thing, sarcomas and sort of bone tumors. And in the end, it's always important to have sort of a multidisciplinary approach to diseases like this one. Um, you get together someone like me who's very passionate about being able to make that diagnosis and be correct every time. This is what it is. I know that's what it is. And I hand off to sort of my multidisciplinary team, which includes a surgeon who says, no, probably shouldn't operate. They're not going to be able to walk well again, if that's the case. Someone like Dr. Wilkie who says, okay, well, if you don't want to operate, I'll get these 5 drugs we could try. These ones work better in my experience, or whatever her experience is, right? She knows the medical side of things. I like this drug, patients like it better. The imaging person will say, okay, great, we'll track it, and they can see it shrinking or something. And so it's like the team spirited approach is incredibly important, I think, in medicine, specifically when you get to rare stuff like this. You really want to have your sort of situation reviewed in a multidisciplinary, specialized center if you can. It's obviously not always possible, and not everybody's cases are complicated, right? If you have a localized tumor, it's excised, it never comes back, this might not be what you need. But certainly for the diffuse type ones, especially the ones that are much more problematic to the patient or to you guys. And then you get the people like that get all together and we all write these giant sort of summary papers about what we think is best. That way, across the country, hopefully you get a somewhat similar treatment every time you go to one of these places. I'm going to save a lot of time for questions. Um, So my summary is that, TCGTs are very odd tumors. They're comprised almost entirely of reactive cells, and that's just an odd thing. That's not the way most tumors form in our bodies. Although they have these recurrent genetic abnormalities, in this case, this CSF1 fusion, I almost never test for it. I can diagnose a tenosynovial giant cell tumor in just a couple seconds under a microscope. I don't need that testing. Um, but it's there. We know it exists. We know these pathways are activated. If your tumor gets weird or if there's something odd about it, we might do that testing. Um, Say you had it diagnosed in a different country and those slides were destroyed, and all I get is a very tiny sample or something like that. I might test it just because I can't see a lot of it, or I might not know. These lesions exist on a spectrum of clinical behavior, and so a tenosynovial giant cell tumor can be incredibly benign, especially the very tiny localized ones. They form as a bump, they're slow growing, they get taken off, they don't come back. Or they can be incredibly aggressive, and I would liken them to a localized sort of malignant process. They're not technically that because they do not metastasize, um, but they grow and they destroy and they cause a lot of harm. And so, in my brain, I think of them sort of in that category, even though technically they would be considered a locally aggressive, I guess, benign process, technically. Pathology plays a key role in diagnosing these things, most commonly with referral up the chain to someone like me. Eventually, you get to a subspecialty center with people that see these all the time, and I diagnose them very easily. It's easier to say that because I see a lot of them. A private practice doctor that sees one of these as a biopsy might never have seen one in their career. I might see six, seven, eight, 10 a month. And so it just depends on your background, your experience. And so, my job is not better than the private guy. We just have different roles. Um, I will say that when someone suspects a tumor is a tenosynovial giant cell tumor and it isn't that, the diagnosis gets complicated very fast, and you definitely want somebody like me in a center like this looking at it. Um, the differential diagnosis, that's what we call like the list of things it could be. If someone thinks something's going to be a TCGT and it gets a sample and it's not, the differential diagnosis includes hundreds and hundreds of other tumor types, some of which are benign and some of which are horribly malignant. And so it gets really complicated really fast, but luckily we don't need that most of the time. All right. So I will stop there and I will take any questions in any form anybody has about anything that I can answer.
Yes, there are several questions people have already submitted, and then other questions I had that have been submitted in the last few days for the recording. One being from a patient here who has a very complicated situation, but they're asking how could they potentially advocate for themselves about having repeat scans or just better care even, and to give some background, this patient is in non-US country, so referral is a key factor in that, and so
Yeah,
looking for some ways and advocating for themselves for continued care.
that is a very hard question. Um, this whole process I've just laid out to you is a very specific process to um, sort of modernized medical environments. We in the U.S. have a lot of resources for medicine. We obviously pay a horrible amount for it, and that's a different problem. Um, but these systems are in place, and so getting referrals is not all that difficult. On a person by person basis, I would say that it never hurts to reach out to people like support groups like this. Groups like this have resources, they've got contacts, they might be able to put you in contact with a clinician who has a study or an institution that loves to give second opinions because they're trying to get more patients to enroll with their drug or something. But I think groups like this one are a good place to go as a general rule. Um, Past that, I'm not super sure. I'm good at the game in the US. It's very hard, and I'm sympathetic to patients that have a hard time with that, especially with something that's classified as a benign disease that I know is not benign to patients. So that I think that can be difficult. I'm sympathetic.
Yeah, we appreciate that. And the patient knows that we will always be there to try and help. But we appreciate any sort of navigation that you guys bring. And I know it gets more intricate the more rural you get, the more different countries have specific health systems. And so understanding that that plays a big role. We did have another question related to that TGCT can be almost entirely reactive, right? What does that mean in regards to symptoms? Does that have any biological plausibility for being more inflammatory or any
Yeah,
clinically meaningful, I guess is the term for things.
yeah, I would say definitely. Most tumors that are benign, most benign neoplasms. they grow, and your symptoms are from that. The tumor gets bigger, it pinches something off, or it makes it so something doesn't work well, but it's all because this thing is just doing this. It's getting bigger and it's messing stuff up where it is. From like a physics standpoint, it's pushing on things. TCGTs are odd in that they activate this inflammatory pathway. They recruit inflammatory cells, and the cells don't just like show up and like hang out. They come to do their thing. They're like, oh God, there's something going on. I'm being recruited to the joint. Everybody go, go. And they all flood that spot and they do what they're going to do. They're like, okay, what are we doing? Oh, fighting infection. Like, all the cells, let their granules go and all the, inflammation gets activated and your tissues get, irritated and red. And so I think that the inflammatory part of this disease probably causes, I mean, I would say it causes all of the symptoms. The tumor cells themselves are so far and few between that they're not big enough to do things in most cases. I think the inflammation that gets brought in, and the giant party that they throw in whatever joint you're in causes all the problems. I mean, sometimes you get very monotonous tumors that are very tumor heavy, and you get more of that classic, this is just growing as a bulk thing that's disrupting things. But I think most of the problem is from this reactive process that gets kicked off. And again, everybody's different. Um, but all the cells that are getting recruited here are coming to do something. They're not just sort of coming to form a mass ball. They're coming to do what they normally would do, which is, destroy tissues or, gobble up inflammation or whatever. And all of that stuff brings redness and heat and all those sort of unpleasant symptoms, I think. It's complicated. It's a very unusual disease process.
I think complicated is probably something that's in both pathologists and I'm a molecular biologist, so in our definition and constant repertoire is how complicated these systems can get. So thank you for that answer. A patient also wanted to know how likely is it that biopsies would not actually capture TGCT cells, and how often do you see even prior samples that may not actually capture it and requiring to have a second biopsy to confirm diagnosis?
Yeah, that's a good question. So I'm still sharing my screen, right?
Yes.
Cool. Let me just blast back up here. So it, I mentioned this gun gets put up here and this needle gets like blasted through the tissue. The person performing this can't see inside your leg or your arm or whatever. There's imaging studies that can help guide them, but they have to use their skill and their judgment and their craft to hit this thing. Because you might have a giant nerve coming right here that they don't want to hit because it will cause problems, or a giant blood vessel they don't want to hit. Maybe it's a really tiny joint. And so missing the sample or missing the tumor is a real thing that happens every day. I sign out, I don't know, probably 10% of the cases that come to me as insufficient or you missed it. Not in this disease specific situation, but it's hard. It's hard to get a good sample. And so I would say the pathologists are pretty good at that. If the clinical team says, "Hey, we have this big bump in a patient and we're going to biopsy it. We think it might be a TCGT," and I get a bunch of fat, right? Imagine this needle could just be like a little bit over here and just go past it and get a bunch of fat. I will say, "I see fat. You missed it. You're going to have to go back." Um, and so that happens. That happens, sometimes. It depends on, where you get the biopsy done, how good they are, how many times they've done this today, what time of the day it is. Is it, it's complicated. How big your leg is, right? Like are you super trim and there's a big bump, or are you a larger person and they have to sort of find their way to this thing? Those things are hard. And I would say most of the time the clinical teams are really good. Most of the time, these tumors tend to be sort of hard when you push on them, and so most of the time it's pretty easy to get a needle going through the thing you care about. Um, but everybody's different. Your body's different. The person doing the biopsy is different, and so sometimes you will unfortunately need another biopsy. And all I can say is, if the clinical team wants another sample and you thought it was worth getting it the first time and you got sort of a bad result and you're not sure, I would get a second one. I mean, I think sometimes you have to try multiple times to make sure you get a good hard piece. Um,
To also appreciate the complexity of that one, like the type of needle that's used, how many passes it takes. It can also be an important factor in making sure that someone's our sarcoma specialist so they know what type
yeah,
of needle and how often to go
for
through it.
sure. Yeah, I mean, some of these needles are really tiny. Imagine like getting your ear pierced, right? They use like a very similar gun. Sometimes you get a super tiny hole. If you get a super tiny needle, why do we do that? We do that because it hurts you less, right? We don't want to cause pain, right? So smaller needle is good. but it gives you smaller tissue for someone like me. We put it in that wax and we cut it. If it's too tiny, it's going to just get shredded up and I'm not going to see anything. And so size of needles, placement of needles, institutional awareness, like all that stuff is really variable. Um,
For someone asking for clarification, the CSF abnormality is only in the tumor tissue, correct? It's not a whole genome, it's not a whole person issue, right? It's just within the.
Correct. Yep. So when we talk about molecular stuff, mutations, whatever, we divide them into somatic and constitutional. Constitutional is germline. You're born with this thing. It causes all sorts of stuff. Somatic, you have a tumor and it just happens to have this thing. Most of the time, tumors have somatic mutations. And so in this case, it's a somatic mutation. Um, just those select tumor cells in your tumor have this thing going on, and the rest of your body doesn't. That's why, theoretically, if you get something like this and you cut it all out, every cell that had that thing is gone, which is why it doesn't recur.
So that's really helpful, and I think sets up the next question. So I'm glad. Thank you for setting me up nicely. What pathological class characteristics, like maybe the percentage of CSF1 receptor aberration or like the production of that abnormality, would that affect the responsiveness to treatment? No, that we
Hmm.
know.
I would say I don't know. That's not my area of expertise, but I can guess based on other tumor types I know and things like that. Um, the drugs specifically target those molecular things, right? Like it's either catching the baseball, blocking the glove, whatever. Um, if you've got more baseballs flying through the air, I imagine the drugs would be acting in a different way. And so for most tumors that have a molecular underpinning, um, those things can be expressed at different levels. Sometimes they're very aggressive and have a ton of the signal. Sometimes they don't have a lot of the signal. Sometimes you have a lot of tumor cells, so therefore a lot of the signal. And sometimes you don't have a lot of tumor cells, especially in this case. And so I think the answer would be yes. I I don't know of a way that we measure like how much CSF is actually happening. I'm sure somebody does that. But I would imagine the level at which this pathway is messed up would correlate pretty well with people responding to the different drugs. That would be just my guess because it would make sense. But that's
And we
that's more
know
of you. That's more you than me.
We know people with 2% versus 16% still respond to the drugs. So you're right. It may even just be that there's a binary yes or no abnormality, and that seems to be responsive. I would say that that may also play a role in how much response is how many tumor cells, right? Once these tumor cells are shrunk or gone or eliminated because their signal is blocked, you're still left with that reactive tissue. So how much is actually
Super, yeah.
left?
Yeah,
Is
I mean that's a hard one. I mean, all cells die, and so I think it might take a while for some of these things to clean up. Even when you have a perfect response and a perfect blockade, all that stuff's still there. Party's still going on, right? They showed up. I mean, all that stuff's going to continue until that process has sort of wound down. But it's a complicated. It's a definitely a complicated sort of cycle. Um, but a lot of people are researching it because scientifically, it's kind of this is probably not a great thing to say, but it's kind of cool that we know how this works. It's
it
very well
rare?
documented.
That's why, yeah.
Yeah. And so, I mean, the fact that there's like 7 drugs or something on that diagram I showed is sort of insane for how rare this disease is. But we know patients suffer from it a lot. And so there's a lot of interest to get something useful because the drug companies know if they get that golden bullet and it fixes this, everybody would take it because the disease can be so bad. And so I think it's a good bad. It's has interesting enough biology that there's a lot of scientific attention toward it. Um, yeah,
Do you know any similar diseases that have the CSF abnormality as like a hallmark? We call things hormonal for those. There's plethora of things that are probably wrong in these tumors. Like they can have more than one trisomy, so they can have more than one chromosome and all sorts of random things. But as Dr. Clay said, the one thing that we care about that drives the growth is the CSF1 aberration. But do we know other diseases that has been essentially the key driving
yeah.
factor?
Um, I don't know of any other tumors that have it. I think that there are some constitutional diseases where like you don't have proper functioning CSF1. It's like the other direction. You don't have enough of it, and I think those people have problems with their immune system and sort of bone structure and things like that. So I think it can go the other way. I think most of those are more again, constitutional or germline or you're born with it. But I don't know of any other tumors that specifically upregulate this pathway. Um,
And that also means those would be system like so if something's germline, it's also often systemic, which means it's everywhere. This one's only
yeah,
local to the joint, right? So someone doesn't have too much CSF everywhere. It just may be just in that joint, right?
yeah, for sure.
Um, we also had if there's any molecular or pathologic features that are predictive of recurrence, and I think you kind of sprinkled
Yeah,
some of those.
yeah. So I mean, the biggest risk of recurrence is not getting it all out, and so we call that margin status. And so, if we go back to dun, dun, dun,
Where's that big honkin' tumor?
yeah, if we go back to this thing, right? Your surgeon's cutting this out of you. They don't want to cut through your muscle. They don't want to hit a giant nerve over here. It's going to make your leg not work for the rest of your life. They're going to do the best they can, but I can't tell where this thing starts and stops, and I don't have to do it inside of your leg while it's bleeding in the OR. And so margin status, I think, is the number one risk factor for local recurrence. Did the whole thing come out? And the pathologist will always outline margins in their reports. Um, I think if you've had a lot of recurrences, sometimes these things can sort of like get out. Like you have a complicated joint and it's recurred a few times and there's a bunch of little bumps now. I think once it sort of has spread around a little bit, the risk of recurrence is just much higher because there's a lot of little tiny parties that might pop up. Um, but margins is the biggest one, especially in the beginning of treatment. But the surgeon might not be able to give you a negative margin no matter what, right? Like they could cut your leg off. Up here, there's normal tissue, right? But we don't want that. That's way worse than the disease. And so they might go into this knowing there's not a chance they're going to get a negative margin. And that often happens with the diffuse subtype. It's just there's no way around it. The synovium is weird. It's very deep. And there's a lot of structures that are super important. And so sometimes just get as much as you can, try drugs, wait for it to recur, go back and do the same process is sometimes all you can do. Like, depending on your biology and your body, there might not be a surgeon on the planet that'd be able to go in and give you a negative margin off the bat. That's just unfortunately part of the process here. So that's important to know. I
That
mean,
commonly with sarcoma and some of the other diseases too, that you just don't want to cut through it. You want to, if you can't remove it, then you try other options sometimes.
yeah, you don't want the treatment to be worse than the thing you're treating. That's always what the oncology, surgeons are telling us. And these things are in a bad location in the body most of the time, and the treatment surgically is often just way worse than what you're getting out of it. It's hard.
Is there anything that you know of within the joint microenvironment that would increase the joint damage or worsen the disease itself? You talked about osteoblast or osteoclast cells.
Yeah, I would say I don't know apart from what I've said. You might know more than me. What do you think?
I think a lot of people are wanting to know what is a predictor of worse outcomes and prognosis, and I think that the we could only guess based off of clinical presentation on like whether there's bone erosion or not. But I
Yeah,
think there's speculated. There's papers that even classify whether there's tendon involvement, nerve entrapment, and like other features that could be, but I don't know
yeah,
molecularly. So I was actually
yeah.
curious.
I think a lot of it's structural. I mean, maybe levels of expression or something, and degree of infiltrating whatever cell subtype might be correlated someday. Um, but it's such a difficult disease to get around. I think a lot of it is local, regional.
And I'm not entirely sure also how you'd get that information for it to be meaningful too, is the other. Are we just like draining joints and seeing what's like I don't
Yeah,
also know
yeah,
like how to clinically put that in. That's going to be someone else's problem.
yeah. I think so too.
Well, thank you so much for spending your time with us, and we absolutely appreciate it. We do, for those interested, have a webinar tomorrow, kind of a back-to-back, which is a roundtable discussion with Dr. Bill Tapp and Richard Rydell to go through how to navigate the discussion of multiple drugs in the space. Dr. Clay nicely
Hmm.
laid out that there's many at this point, and there will
Yeah.
continue to be. So how we make those decisions, how providers consult patients and considerations that you should potentially discuss when deciding what to go on. That will be tomorrow at 11 a.m. Eastern Standard Time. And thank you all very, very much. And thank you especially, Dr. Clay.
Of course, yeah. If anybody has questions, Sydney can get a hold of me. I'm always happy to try my best from the the minimal amount of perspective that I can bring to bear.
Oh no, you're wonderful, and you're getting claps and hearts. So
All
thank you.
right. Thanks for your attention, everybody. Good luck with everything.
Bye.